Okay, so this is chapter 3, part 1, and picking up at slide 27. So we're just about to start describing the membranous organelles. So the 5 types of membranous organelles would be endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria. So the first category, endoplasmic reticulum, is actually 2. So what it is actually a continuation of a structure we're going to learn about a little bit later called the nuclear envelope. So it's a double membrane structure that surrounds the nucleus is the nuclear envelope, and then it continues on kind of folding back and forth out in the cytoplasm, and it's going to be divided into this smooth version and the rough version. Between the two of them, their jobs are to create proteins, carbohydrates, and lipids, to store the things that they create, to transport them around within the endoplasmic reticulum, and especially the smooth ER is responsible for detoxification of drugs or toxins. So again, smooth ER is called smooth because we do not have any ribosomes on it. Fixed ribosomes on the surface is what makes the rough ER rough. Now, since ribosomes are required for making proteins, then you know that there are no proteins being made in the smooth endoplasmic reticulum because it doesn't have any ribosomes. Instead, it's going to make the lipids and carbohydrates for the cell. So remember, proteins, lipids, and carbohydrates are the 3 biggies, right, of the macromolecules, those with nucleic acids. So the things that the smooth ER makes: phospholipids and cholesterol, steroid hormones, glycerides, and glycogen. So you can see right there, it's the lipids and carbohydrates that are being made by the smooth ER. The rough ER is covered with ribosomes, so it makes it look like it would have a rough surface. And so these are going to be protein factories that are creating proteins that are going to leave the cell. So we're going to make proteins and glycoproteins. We're going to fold them up to their secondary, tertiary structures within the rough ER. And we're going to package them into what are called transport vesicles and send them over to the Golgi apparatus. So anytime you make a bubble out of a membrane inside a cell, it's called a vesicle. And unless we tell you what the vesicle is doing or what's in the vesicle, you really couldn't tell a difference. Okay, so we have to kind of give you a little bit more information about the vesicles. This one is a transport vesicle because it's transporting from the rough ER over to the Golgi apparatus. So here's a diagram. So over here you see here is the nuclear envelope. It's kind of a little bit darker blue. And it just kind of has these little branches that go off. And then it folds back and forth. And so you see some places we see these darker circles. Those would be the fixed ribosomes making this rough ER. And then out here at the fringes, it changes shape and gets more pipey, less flat. And this would be the smooth endoplasmic reticulum. Make sure you learn how to spell that. You will not be allowed to abbreviate it as ER on anything, okay? So make sure you can spell endoplasmic reticulum. This would be the rough endoplasmic reticulum covered with ribosomes, fixed ribosomes. This is the smooth endoplasmic reticulum with no ribosomes. The next organelle is the Golgi apparatus. Now, things are passed through the Golgi. I like to think of Golgi as the UPS guy. It finishes packaging them into their final vesicle, and then it delivers them. Sometimes the Golgi will send the vesicle into the cell, and the cell will hold on to it for a while. Sometimes it's going to be passed up towards the surface of the plasma membrane to be secreted. So then that will become a secretory vesicle. So jobs, what's going on inside the Golgi? It's going to modify and package secretions and get them ready for release. It can add or remove carbohydrates from the proteins that were created by the rough ER. It is going to be responsible for making new sections of membrane that are just going to be, when the vesicle goes to the surface, it's just going to open itself up and join the membrane. So we're renewing sections of the membrane from the walls of vesicles. It can package special enzymes within vesicles, and they will become lysosomes in order to be used by the cell as a separate organelle. So here is a picture of the Golgi apparatus. So things would be brought into this bottom side in transport vesicles, and the Golgi is basically these long flattened sacs called cisternae. And we're going to package up the proteins and modify them, and then when they come out the other side of the Golgi towards the outside of the cell, they're going to be in secretory vesicles or in vesicles that are going to be passed inside the cell. Lysosomes. So if the Golgi takes a bunch of very strong enzymes, and remember enzymes are proteins, and packages them into a vesicle and the cell keeps it, then it can become a lysosome. Now, as long as that package of enzymes has not seen any of the substrates of those enzymes, it is called a primary lysosome, and the enzymes inside will be inactive. Now, when it wants to use a lysosome, it will put the substrate that it gets from outside the cell into another vesicle through a process we're going to learn in this chapter called endocytosis. And then it will bring the vesicle with the substrate to the vesicle that is the lysosome, and they will merge into one giant vesicle. That will then be called a secondary lysosome, and the enzymes inside it will become active. They will activate, and then they will digest whatever we brought in that second vesicle. So we use these to destroy bacteria, break down molecules ingested by the cell, or we can even use it to recycle a damaged old organelle if we want to rebuild it and use the parts. Now, a cell can also have a special triggered suicide mode. It's called autolysis or apoptosis. And if it is told to commit suicide, the lysosomal membrane of the inactive lysosome will just break open and release the enzymes into the cell. Now, once those enzymes are released, they will see substrates and they will become active. And so the cell digests itself from the inside out. And all cellular materials are recycled and reabsorbed by nearby cells. So apoptosis or programmed cell death is actually a very neat and tidy, planned death. As opposed to another type of cell death, which is called necrosis, which is caused when a cell gets just ripped or torn or burned or damaged with acid. It's an unplanned death, and instead the cell contents just leak out all over the place with no chance to recycle them. So necrosis is a bad cell death. Apoptosis is a good cell death. Peroxisomes are kind of like lysosomes, and in that they have enzymes in them. The difference is that the enzymes in there are going to break down organic compounds such as fatty acids, and they're going to produce peroxide as a byproduct. And so they also contain another enzyme called catalase, because peroxide would be toxic to the cell. So the catalase that's also included in the peroxisomes is able to take hydrogen peroxide and convert it to oxygen and water and make it safe for the cell. So peroxisomes are going to break down peroxide, and it's going to break down products that produce peroxide in their breaking down, if that makes any sense. All right, mitochondria. Now that's the plural form. That means more than one. If you only have one, it is a mitochondrion. Now these are actually cool organelles. They have a couple of really neat things about them. Number one, they have two membranes, and the membrane on the inside is bigger than the membrane on the outside. So imagine you had like a gallon-size Ziploc bag and you had a quart-size Ziploc bag, and you stick the gallon-size one inside the quart one. What happens? Well, you have to wrinkle up the gallon-size bag. So when you stick this larger inner membrane, it forms wrinkles or folds called cristae. And then in the very center would be the matrix. Now the job of the mitochondria is to take our food energy, things like glucose, fructose, all those, and we're going to turn it into adenosine triphosphate or ATP by using a process called aerobic metabolism or cellular respiration. This is how we get 95% of the energy that the cell needs to stay alive. And just to really break it down, and I would not ask you much about this on the test, just the basics is that we're going to take sugar and water and ADP, and after we finish with cellular respiration, we're going to have created glucose and oxygen in ATP, and we're going to create carbon dioxide, water, and ATP as products. As we get into anatomy 2, you'll learn a lot more about all the individual processes that happen inside the mitochondria and just outside the mitochondria to create this ATP. But this is what a mitochondrion looks like. So you can see, because the outer membrane, or excuse me, the inner membrane is larger than the outer membrane, it is wavy. So anytime you see a cell model and when they cut into it, you see this waviness inside, you know you are looking at a mitochondrion. And the enzymes are all studded on these inner membrane walls towards the matrix side, and a few towards the inner membrane space. All right, I'm going to stop this one, and we'll pick up with the nucleus in the next video.